Good afternoon. My name is David Pauly, and I am honored to have been the 2025 recipient of the Rolfe Medal for Excellence in Morphometrics, given at Stony Brook, New York, Stony Brook University, by James Rolfe himself on October 22nd, 2025, just a few days before his birthday. At that event, I gave a talk which was inadvertently not recorded, so I am recreating it here. At the event, Stony Brook President Andrea Goldsmith gave some introductory remarks about the medal, and to me, Anthropology Chair Kathy Twiss also gave some introductory remarks about both the medal and myself. Jim Rolfe himself got up to talk a little bit about the medal and about morphometrics, and Eric Delson, who was the chair of the award committee, then introduced me and said lots of nice things about me. I was really pleased to have heard from Eric earlier in the summer telling me I got the medal. And in fact, when I got off the phone with him, my wife, who'd been in the other room, said, "Who was that?" It sounded like it was the MacArthur Foundation calling to give you an award. And I said, well, it wasn't the MacArthur Foundation, but it was the next best thing. It was Eric Delson saying that I was to receive the Rolfe Medal. And I'm really honored to have received that medal, especially if you look at the people who have received it before: Fred Bookstein, Paul O'Higgins, Benedict Hallgrimson, Dennis Slice, Dean Adams, Joan Rixmeyer, Philippe Mitterracher. These are all people who did foundational work in geometric morphometrics, and in many cases in morphometrics in general. And all of them are people who I have learned from, some of whom I've worked with, and all of whom I look up to and respect as people who can answer my questions and teach me things. And so I'm really deeply honored to follow in their footsteps. My talk this afternoon is about the interaction between phylogeny and morphometrics, which is of great interest today. A lot of morphometric work involves phylogenetic comparative methods, and from my earliest days dealing with morphometrics, it's a topic that I have been interested in. And specifically in this talk, I'm going to share with you things that worry me about phylogeny and morphometrics, and I'll explain exactly what that means. But what I want to do is pose some questions about whether it is always possible to estimate things like evolutionary transitions, ancestor state reconstructions, phenotypic change on a phylogeny, rates or modes of evolution, adaptive landscapes. These are all things that today we do routinely. I do them in my own research, and many papers are published today that use morphometrics in this way. And what I really want to ask is, in what ways do evolutionary paths map onto mathematical paths? And in this talk, I'm going to give a little bit of background on exactly what a morphospace is, different ways in which there might be empty gaps in morphospace, and what it would mean to trace trajectories across them, an evolutionary type trajectory or a phylogenetic trajectory. And in just about every morphometric study that's done in any respect, we estimate some sort of path through the morphospace defined by the variables in that study. That may be a very simple in very simple ways, like on the far left, which is just simply a bivariate plot, which is probably the simplest sort of morphometrics you can have, looking at ranges of variation in teeth, in this case, looking for gaps between them, and diagnosing, in my case, in this study, trying to diagnose fossil species based on the sorts of gaps and the sort of variation that we see in living species. You might also trace evolutionary trajectories, and I don't want to pick on anyone else's work because I'm going to question some of these things, so I'm picking on my own. But like in this 2008 paper that I wrote on the evolution of tarsals, and adaptive peaks, tracing an evolutionary trajectory from an ancestor of seals to a living seal. We might estimate paths in morphospace through modeling and simulations. This is a hypothetical example from a paper that I wrote for evolutionary biology, actually at the request of Benedict Hall-Grimson, looking at how a trajectory in genetic space might map onto a trajectory in morphospace, in geometric morphometric morphospace. Or ancestral reconstruction, which is also very commonly done. A paper I worked on with Ida Gomez in 2013, where we used ancestral state reconstruction and some of the uncertainties that come from the Brownian motion process around that to create confidence intervals around those reconstructions, to then ask whether fossils that are found in the hominin record might or might not actually be probable as an ancestor. And all of those uses, which again are very common, one way or the other, we're estimating a path. We're tracing a line between two objects which we can actually measure, and saying something about the intermediate space that evolution may have gone through it, an ancestor may have occupied it, etc. And even in the very simplest sorts of morphometric exercises, like taking morphometric measurements on a sample of a single species or population and estimating the mean of that population, the population mean occupies a space in that morphospace that is connected by the shortest paths to all of the members of our samples. So paths through morphospace are just a fundamental part of geometric morphometrics, or any kind of morphometrics for that matter. And what I want to do is look at them more closely, think about the construction of morphospaces, think about what it means to draw a line through them, and think about situations where that line may be really quite well justified and we can have a lot of confidence in it, versus situations where we might want to think twice about it. So first, let me just review some of the mathematics and concepts behind a geometric morphometric morphospace, where a morphospace is really all that means is it's a mathematical space that's defined by the variables that go into the analysis. In geometric morphometrics, those are going to be landmark coordinates. In some sort of other morphometrics, they might be measurements or what have you. And the number of dimensions in that space is defined by the number of variables and the transformations to those variables, which may or may not remove degrees of freedom, which they certainly do in geometric morphometrics. But generally speaking, a morphospace, especially a multidimensional one, we often view it as a principal component space, where the center of that space is centered on the sample's mean, where the first principal component follows the greatest range of variance, which is usually the greatest difference in shapes. The second principal component is orthogonal to that, and so on. And our individual objects are spaced in that space. And here we see some modeled shells, various types. And things about this, the first, any of these axes we can describe as a shape spectrum. In geometric morphometrics especially, we often don't look at the numbers per se. We look at models that show how the shape changes from one side of that axis to another. And there's an axis for each one of the many dimensions of the morphospace. The objects in that morphospace have what are called scores, which is just simply the XY coordinates in that space. So the score of Dalmanella here on PC1 is 0.42. On PC2, it's 0.20. If we were down in this quadrant, they would be negative numbers, which means they're on one side or the other of the mean. You can calculate Euclidean distances between the objects in that morphospace fairly simply. This is calculated in just the first two dimensions, but you can calculate the distance across all of the dimensions. And one important component of most geometric morphometric studies, if the data have been transformed right, what we're looking for is a PCA space in which the distance between the objects in that PCA space is identical to the distance between their landmarks when they have been Procrustes superimposed. So if we took these two shells, Ilmatogyra and Poromya, and Procrustes superimposed them and then looked at the distance between all the corresponding landmarks, which are shown here as displacement vectors, added all those up, that would be what we would define as the Procrustes distance between them, the sum of displacements between the homologous landmarks after Procrustes superimposition. If we calculate the distance between those two taxa in the PCA space, and presuming we calculate this distance across all the PCA space, the number will be identical if one has constructed that space appropriately. And this is an important property of morphometric spaces in all sorts of ways. And one of the things it means is that in a morphometric morphospace, the distances between objects are preserved. So if you have two objects, put them into the morphospace, that distance is exactly the same as their landmarks, and that means that it is not sample dependent. But geometric morphometric morphospaces are very strongly morphometric. We may have dozens, if not thousands, of variables, and that makes the principal component space very highly multidimensional. So the distances between the objects in that space are constant, but the principal component's axes themselves are sample dependent, and that's because they are centered on the sample mean. So that the sample itself defines where the origin of that the coordinate space is, and also because the first axis traverses the major line of variance. Then if we add or subtract something from that sample, we'll change the variance and change the first line. And you can see that in this video over here. In this case, what's happening is the red object is being added and subtracted. You're seeing what happens to the major axes, or the relationships between the objects in the full morphospace, which in this case is just 3 dimensions. The distances between those objects don't change, but the principal component axes change. And what that means is that when you're only looking at a small number of principal components, like the first and second principal component, their relationships appear to change, but that is just simply because the projection is changing, your view of that. So the addition of a new specimen, it simply changes the coordinate system. It doesn't change the relationship between the shapes that were common between before and after the new object was added. So in other words, the principal component axes are sample dependent, and that means that statistical operations or reconstruction operations on just a small number of PCs are sample dependent. And the patterns of interest, including phylogenetic patterns, may or may not be represented on the first principal component axes. That's always something to wonder about if you're doing a morphometric study. Whereas operations, statistical tests, or what have you, carried out in the full multivariate morphospace are not sample dependent in the same way that they would be if you did a variable reduction. And that's something to keep in mind as we go through the rest of this, and you'll see why that matters later. My career, I started off interested in phylogenetics, particularly in cladistics and parsimony, which I got interested in as an undergraduate when I worked with Tim Rowe at University of Texas at Austin. I took a class with Tim. It was my first paleontology class. It was highly phylogenetic. I was interested before that class in computers and in programming. And Tim would talk about doing phylogenetic analysis. He'd say we learned about the characteristics of different groups and the features that they shared. And he said you would take these data and put it into the computer and you get out a tree. And having done some programming, I had no idea, I couldn't even imagine how that was done. So I asked him if I could do a project with him, which ended up being phylogeny of some mammalian carnivores. And my interest in phylogenetics has carried with me through my career. And when I started working with morphometrics, that was one of the major interactions. Our major interest on my part is the interaction between morphometrics and phylogenetics. Phylogenetics, of course, drawing trees, is drawing connections between tip taxa. So if we're thinking about tip taxa in a morphospace, then what we're thinking about in reconstructing a phylogenetic tree is in fact drawing lines, making trajectories between points in a space, reconstructing ancestors perhaps, calculating rates of change across those branches. Phylogenetics has come a long way, and computational phylogeny reconstruction from morphology has also had a long history. Started off probably actually in the late 50s, but really getting going with real algorithms in the 1960s, like the Cavalli-Sforza and Edwards paper shown here. Discrete traits and parsimony, computational methods for them came along actually a little bit later than for continuous traits, because continuous traits and gene frequencies are very similar in certain respects. And then Felsenstein in 1973 with likelihood method for constructing trees from continuous traits. It was later, after I'd gotten my first faculty job at the University of London at the Queen Mary campus, that I really started learning about morphometrics. I'd been a postdoc at University of Michigan, where with Miriam Zeldich and to some extent with Fred Bookstein, had started to get interested in morphometrics, and then moved to London and worked a lot with Norman MacLeod, where I really started to learn about morphometrics both as a user and as a programmer. And the very first talk I gave on morphometrics was at this 2002 Systematics Association annual meeting that Norm and Peter Forey convened to talk about morphometrics and phylogeny. And you may be aware that in the formative days, particularly in the 70s, 80s, and into the 90s a little bit, there was a long-standing acrimony, if you will, between people performing parsimony-based phylogenetics and people who were working on morphometrics of continuous traits, which focused on whether you were only using shared derived traits to construct a tree, or whether you were using quote-unquote overall similarity. And so there had been long debates, and some of that was dying down a little bit by the early 2000s, which really kind of opened the door for new ways of thinking about this. And this symposium was convened for exactly that reason. And you can see some of the people who were there. Fred Bookstein was there, but also, and Jim Rolfe, but also Joe Felsenstein, Mark Pagel, Andy Purvis, and others. Were in attendance, participating, giving talks here. And my very first morphometric talk was at this symposium. And you can imagine being a beginner, this was somewhat intimidating, especially to have Jim Rolfe in the audience. I don't know whether Jim listened to my talk then. In some ways, I sort of hope he didn't. But today, or on October 22nd, where again I was giving a talk in front of Jim Rolfe about morphometrics, it's still intimidating. And these people are very accomplished. And it was a really great symposium. One of the interesting things at that symposium, Jim Rolfe's own paper was about projecting phylogenetic trees into a geometric morphometric morphospace, what we would today is often referred to as a phylomorphospace. And to the best of my knowledge, this was the first real fusion of geometric morphometrics and what we would now call statistical phylogenetics. And what I mean by statistical phylogenetics is any sort of probabilistic likelihood or Bayesian model for the relationships between characters in trees, or in this case between variables in trees, or the geometric Cartesian coordinates of geometric morphometric shape. And statistical phylogenetics is used for everything from phylogeny reconstruction, ancestor state reconstruction, or ancestor shape reconstruction, estimating evolutionary rates, model selection for evolutionary modes, hypothesis testing for rate and mode shifts, testing for evolutionary convergence, testing for multiple adaptive peaks, performance trade-off modeling, etc. This is really where the state of the art in evolutionary geometric morphometrics is today. And it had really been getting going there in the very late 1990s and early 2000s. This paper that papers that you can see up here, the book by Harvey and Pagel, had come out on the comparative method in evolutionary biology, which was not using geometric morphometrics per se, but was using continuous traits. And to me, a paleontologist, the quite provocative works like this one from Paul Harvey, Bob May, and Sean Nee, titled Phylogeny Without Fossils. So I also got quite interested in this because that seemed like quite a challenge to me. Being a paleontologist, I tend to believe that phylogenies or fossils are quite interesting and useful. And in many of these models, Brownian motion is the null model, and most other models are juxtaposed against it. And let's see if I can get this to move without drawing on it. Hello. And because I have the pens, there we go. All right, let's pause. All right. The null model is Brownian motion. Most other models are juxtaposed against it. And here on the left, you can see a typical Brownian motion model of a trait through time, which starts with an ancestral population. Let me get my laser pointer back. Starts with an ancestral population, and at each step it either drifts left or drifts to the right, drifts higher or lower on this trait, and makes a Brownian motion model through this to a descendant population. And genetic or selectional drift are processes that could cause this. Now I need to get my pointer back here. On the right there is what is called an OU, or Ornstein-Uhlenbeck process that has some Brownian motion in it, but there's selection in that process to keep the trait heading toward a certain target, which in this case is the where the ancestor was, but it could be somewhere else. And the selection is always, if it drifts away too far, selection brings it back. Most phylogenetic statistical models are based on Russ Landy's adaptive peak model from his classic 1976 paper, and then developed by himself and others subsequently, which is a model for adaptive selection where you have in quantitative traits, continuous traits, including potentially geometric morphometric traits, where you may have a population here with some sort of a variance P and a mean trait value Z bar. We have an optimum trait value that would maximize fitness, and selection on that is represented as an adaptive peak, here represented as W bar, the average selection on that, where the log of the slope of this is proportional to the amount of selection. And so in evolution, related to this selection and or drift, Landy proposed a formula that delta Z bar, or the change in phenotype at any step, is equal to the selection component here times the genetic component of the phenotypic variance, plus a drift component, which is the genetic variance divided by the population size. So the amount of drift varies with population size, and the degree of selection has to do with the width of this adaptive peak, whether it's shallow or whether it's steep. And the differences between many evolutionary models are basically functions of those two components. A Brownian motion model, selection is effectively zero, and the drift component dominates. And so if you run many of those many times, then you get actually an expected distribution. Most of them, the most likely place to end up is where you started, with decreasing likelihood as you get further away from that. And that distribution is related to the number of steps that have elapsed and the per step rate, which in the Landy model would be the G divided by N. And the Ornstein-Uhlenbeck model is where the adaptive peak predominates, and it has its own center or mean and its own variance, so the expectation of the outcomes is related to those two parameters in the adaptive peak. And you could also have some sort of directional, where you're just sort of selecting things in the same direction, not exactly towards a peak, or put another way, towards a peak that's infinitely far away. And those are three examples of statistical models. And the idea behind them is that each one of them has an expectation for the range of outcomes, from which you can give a likelihood for different outcomes. Is it a likely outcome? Is it an unlikely outcome? And then you can look at real data to ask whether those real data better fit, for example, this model or this model or this model. And the examples I've been giving you are lineages through time. And Gene Hunt's work, he developed this for paleontological data series where they literally you've sampled the same species or the same evolving lineage through a series of paleontological beds, so you have observations at different times. And Hunt, in a series of papers, developed statistical models where you could estimate the rates of evolution, estimate the modes of evolution by asking which of those modes fit. If you're dealing with a statistical model on a phylogenetic tree, it's basically the same idea, except those lineages branch. And so in this case, the expectation has to do with the distribution of several tips that come from the original ancestor, and their tips values are co-varying by the amount of shared ancestry. But the basic statistical principle is the same. Using the phylogenetic topology, the branch links, you can develop a statistical model for what the expectation of the outcome of the tip values would be under Brownian motion or an OU process or any other process that you can imagine. Ancestor reconstruction uses the exact same statistics. If it's using a Brownian motion model, instead of looking at what the expected outcome for descendants is given an ancestor, instead what we're looking at is the expected value for an ancestor given a tip. And in a Brownian motion process, the probabilities going backwards are exactly the same way as coming forwards. So if we know a descendant, the most likely ancestral value is the same value, trait value is the same value as the descendant, with a normal distribution becoming less and less likely, and the variance on that estimate has to do with the rate of evolution of that trait, which of course we don't know a priori. We would have to estimate that too. If you have two or more descendants, then each one of those descendants has a most likely ancestral value and a distribution. But if we know that they're both descended from the same ancestor, then that constrains the most likely ancestor basically to be the sum of those two probability distributions. And so we reconstruct the most likely ancestors based on a tree topology exactly like this. So a phylomorphospace, like the one that Rolf first revealed at that 2002 conference, is based on those models. So using a Brownian motion model, these are some data, human skull, wallaby skull, etc., morphometrics, with ancestral nodes reconstructed and the tree drawn in to them. And of course, each one of those lines and each one of those reconstructions is a type of trajectory through a morphospace that we're reconstructing based on the mathematics of the morphospace and the statistical expectations of the evolutionary model behind it, in this case, a Brownian motion evolutionary model. And I mentioned before, paper I worked on with Ida Gomez Robles on hominin evolution, where some parts of the human fossil record and phylogeny are pretty well agreed on, and other parts of it are less well agreed on. And so in this paper, led by Ida, looked at some of the taxa whose relationships are pretty well agreed upon. And then, based on the tree topology of those, asked the question, what, under a Brownian motion model, what would the most likely ancestral value be, and what is the range of variance around that under a Brownian motion model? We can turn that into a probability distribution or a likelihood distribution, and then looked at putative fossils that might be the ancestor, and then ask what is the likelihood of that particular fossil being an ancestor for that particular node. Again, using those statistics, statistical phylogenetic approaches in a geometric morphometric morphospace to test hypotheses. All well and good. So the question now, getting really to the meat of this presentation rather than the background, we might ask ourselves, do these lines in morphospace represent realistic biological paths? Because under a likelihood model of Brownian motion, the single most likely path through a morphospace is a straight line back to the ancestor or whatever. We expect all sorts of deviations away from it, but each one of those deviations has less probability than a straight line. Basically the same principle shown before with the ancestor reconstruction, where the most likely ancestor is the descendant value, or if it's somewhere else in a straight line between the descendant value. So we could ask this question. We have two of these shells that are observed. We can draw a line between them, which would be the shortest evolutionary path, or the most likely evolutionary path, or the most parsimonious evolutionary path, if you will. We can draw a line through that, and in geometric morphometrics, using the principles described before, where the PCA space is just simply a transformation of the landmark space, we could reconstruct that evolutionary path, and that's what you see in this animation at the right. That would be the shortest evolutionary path between those, and that's exactly what we would expect if we were selecting from one to the other. We're drawing a line through the empty regions of morphospace, but we might ask, is that transition that we see here a realistic one? Is it biologically realistic? Did evolution actually follow that path? And that's what I want to ask here. And to think about this, I've sort of categorized empty areas of morphospace into several types. And I'm going to give them names here. These names are my own. But imagine we have, this is a case of some moles that are, and these, I believe, are the skull, geometric morphometrics on the skulls of moles. These are very closely related taxa. Some of them are subspecies. Some of them are basically cryptic sibling species. This is a paper that I worked on with Islam Gundas and colleagues a few years ago. And each one of these taxa has several individuals in it, so it's measuring something about within subspecies variation and differences between very closely related species. So these are very small geometric morphometric distances, and we can kind of imagine that the spaces between these individuals, there may be other individuals that would fill those spaces, and we can quite reasonably expect those spaces to be filled with some other as yet unsampled mole. So I'm going to call these ordinary gaps. This is the sort of gap we expect. We sort of expect that biology could could put a point down in any one of these locations, any unsampled area. And so we have some reasonable expectation of expecting those gaps to be filled by real biological shapes. Another type of empty morphospace has to do with the mathematics of the topology of these space, what I'm going to call Kendall curvature, because in geometric morphometrics we often talk about Kendall's shape space, which is a curved morphospace. In this case we're looking at a shape space of triangles where it forms a sphere, but if we have more landmark coordinates than just the coordinates of a triangle, it would be a multi-dimensional hypersphere. But in any event, geometric morphospace, because of Procrustes superimposition, is curved. It's not technically a Euclidean morphospace. We often say that biological shape variation is small, and we can often discount this curvature. But that curvature is real, and what it means is there are some parts of space that don't exist, if you will. And you can see this with a simple experiment if you want to. If you generate some random triangles, I've done 1, 2, 3, 4, 5, 6 of them there. And we do geometric morphometrics on them because of the degrees of freedom in Procrustes. There's technically only 2 degrees of freedom in that, so PC1 and PC2 represent all the shape variation in those triangles. This looks like a perfectly ordinary PCA. You don't notice anything strange about it, but if you did that some 1,000 times and generated 1,000 random triangles of all possible shapes and then plotted them in the PCA space, you start to see a pattern here. The corners are not filled, and that's because the corners don't actually exist. We're looking at that as though it were a Euclidean space, but in fact the triangles are constrained by Procrustes superimposition to lie on that sphere. And in fact, even though we expect there not to be a third dimension, if you plot the third principal component of these data, you can see that curvature on space. So all of those points are lying on a curved but two-dimensional surface. And so there is really no way for a triangle to fill these corners or the convex or concave parts of this. They're constrained, and this is a mathematical type constraint, a topology constraint. That means there are some empty parts of this morphospace that really don't exist, and no shape can actually lie there. And again, we often ignore that possibility in geometric morphometrics, but it does exist. The third type of empty area of morphospace is sort of a mathematical interaction, but it's not a constraint per se in the same way. And I'm going to call these Bookstein bins because Bookstein in 2013, this paper called Random Walk as a Null Model, discussed these quite a lot. And these are sort of interesting. These are what some people would refer to as horseshoe shapes. But when you do principal components on a gradient of a variable gradient, and it's a high dimensional variable gradient, then it can do interesting things in a morphospace. So we just looked at a random walk. What I'm showing here on the left is an evolutionary random walk. But instead of just one variable like in the previous slide, this one is actually with 56 variables, sort of like a lowish dimension geometric morphometric analysis with three-dimensional landmarks and just a handful of three-dimensional landmarks. This graph is showing 2 of those, which are represented as x and y against time, and it's just a random walk. But you can imagine this being a 56 dimensional space, that random walk moving randomly through all of those variables. And I've drawn a line here showing where the most likely value is, and we've reached some area off of that. So interestingly, if you take those 56 variables on just that simple random walk and do a principal components on them and just looked at PC1 and PC2, you get this very nice horseshoe shape. And even more bizarrely, if you haven't considered this before, if you do PC2 and PC3, you get this nice loop. Why is that? There's a very simple reason, and that's because we're sampling all the steps of this random walk. And what that means is the two most different objects in that data set are, statistically speaking, the first one and the last one. And so since the first principal component defines the greatest variance, which is basically going to be the greatest difference in that data set is usually the first one versus the last one. PC2 is going to get the next greatest distance, which is going to be the middle one. It's going to be the most different from the two ends. And PC3 is going to pick up the next greatest trajectory of distance, which is basically the quarter marks. And so if you perform random walks many, many times, and this is just an animation of a series of them, each time they form this same sort of horseshoe trajectory. And what that means is that no matter where this thing is going, if we're looking where the random walk is going, it's actually sampling the entire variable space. But if we look at it in just the lowest dimensions of the first and second principal component, it looks like there's space in there. And you can sort of imagine if we drew a trajectory between the start and the end, we're crossing some space where the evolution never went exactly. And this is a really interesting and complex space, and it's mostly a visual artifact of looking at a high-dimensional pattern in low-dimensional space. So going back to that early animation that I showed, the relationships between these objects in principal component 1 and 2 is somewhat distorted from what it would be if we could actually look at it in all of the dimensions. Because if we look at all of the dimensions, it would look just like a random walk. And I first encountered those long before reading Bookstein's paper, but nevertheless was working on a problem with Jason Head on snakes, where we were looking at gradients of shape along vertebrae, which is exactly the same problem. It's not a random walk per se, but where vertebra 1 is very similar to vertebra 2, and vertebra 2 to vertebra 3, and vertebra 3 to vertebra 4, all the way through a column like this. And if you do a principal components on that shape gradient, you also get one of these horseshoes. These are common. There's no variation out here. The variation is curved in an interesting way because of looking at it in low-dimensional space. If you look at an allometric trajectory, where you're looking at a sample of growth from young to older, that's also a gradational series, and so you also see these sort of Bookstein bends, as I'm calling them here. And if you're a paleontologist, you've often worked with phylogenetic trees like this. This is a paper from my undergraduate advisor, Tim Rowe, and Jacques Gauthier, looking at the relationships of extant mammals to various fossil taxa. And because the question of interest was the phylogenetic origin of Mammalia and some of the transitions that went to it, the choice of tip taxa here was what we would call pectinate, looking at a series of outgroups. They weren't interested in the relationships among taxa in any of those particular outgroups. They were looking at the sequence of changes leading to mammals, which creates a pectinate tree. And if we only looked at the morphology on that tree, we would see one of these trajectories. A pectinate tree is more or less going to produce a pattern very much like the random walk itself. So this is a phylogenetic tree where you can see one of those Bookstein bends. In this case, I've randomly generated both the tree, randomly generated a pectinate tree and data on it, but it serves to show the relationships here. And what that means, again, is this empty space in the middle. You could have taxa there, but because of the way the space is constructed on the first two principal components, you appear not to. And that's, again, a methodological artifact, if you will. It's by looking at a high-dimensional space in just 2 dimensions. And then the fourth and final type I want to talk about is a biological type. The first one was biological, we're just not sampling it. Types 2 and types 3, the Kendall type and the Bookstein type, they are mathematical artifacts in some way that create an empty space. This final one is a biological reason why you might have empty morphospace, because it might not biologically be possible to have certain sorts of morphology, for example. And I'm going to call these constraint chasms, and my thinking on this really started reading works like by Per Ahlberg, who had worked on developmental constraint or ontogenetic constraints, and arguing that developmental processes made some sorts of morphological outcomes impossible, at least improbable, if not impossible. This series of diagrams down here at the bottom is basically human aortic arches looking at variations on them, because we have sort of the standard anatomy you would learn in a textbook, but there are all sorts of variants that real people may have, where these the arteries connect into the aorta in different ways. And there's not a continuous variation in it. There's sort of this pattern or this pattern or this pattern, and they're related to one another. And so what Ahlberg argued is that there are some morphological outcomes that development makes easy, and other morphological intermediates that development makes difficult or impossible. And so this is a biological sort of way that we might have gaps in a morphospace, as this cartoon shows. And I'll show you an example using geometric morphometrics that illustrates how this is going to relate back to phylogeny and ancestor reconstruction. And this is really a thought experiment that I came up with. If you were doing some sort of geometric morphometric analysis, and here I'm going to do an outline analysis, and in some ways this is kind of contrived to make a point, but the same principle is going to hold in any real data set that's less contrived. So this is a fossil antilocaprid, Hexobalamerix, which is a real animal. It had sort of 3 horns on it. This one over here is a hypothetical antelope, which has horns on the nose, and I'll show you in a moment that that's not an improbable sort of morphology. And imagine we have these 2 taxa. I'm a paleontologist. I want to know something about their ancestor. I've done some geometric morphometric representations of them as an outline. I ask, what is the last common ancestor? If I don't know any of the rest of the phylogeny, our tree is pretty simple to reconstruct. We've got sister taxa. And if we reconstruct an ancestor under Brownian motion model, that's actually a very simple thing to do. It's technically just the average of it, and you get a reconstruction that looks like this. One big sort of cone back here on the back part of the head, and a pair of horns sort of right here halfway between the eyes and the nose. And if you work on mammals and know something about the variation in horns in artiodactyls, this seems really biologically implausible. Having a big cone-like thing there, but also even just having horns in that position on the forehead is just not something you see. It's very biologically implausible. And you'll see why. Here are a whole bunch of fossil antelope. Some of them do indeed have big horns on the nose. Some of them have horns on the head. Sometimes they're long, sometimes they're short. Sometimes they don't have any. All sorts of variants on that. And if we go to a morphospace, the exercise I just showed you would look like this. Here's those two tip taxa. Our reconstructed ancestor would be here in the middle. The branches would be the shortest distance between them, and we're sampling an area that seems biologically improbable. If we put all of those other fossil taxa in there, you get a pattern that looks like this. They also form a horseshoe, but in this case that horseshoe isn't a mathematical artifact of some sort of a continuous gradational series. Rather, this horseshoe is because there are some areas of this morphospace that are basically biological no-goes. The processes of development and genetics in a mammal just simply don't produce a horn pattern that would correspond to this area of morphospace. And this sort of thing probably exists. It is much harder to document biological places where evolution can't get or development can't get, because we can always wonder, well, maybe it just didn't happen. We haven't sampled that. One of the best examples showing that this may well be the case is a paper I like to refer to by Isaac Salazar Ciudad and Yuka Yarnval on teeth, where they know a lot about the development of teeth. They worked out many of the molecular mechanisms that go into the formation of the morphology. Isaac programmed a computational model to take those molecular parameters, turn them into a cellular level developmental process where you could tweak those underlying genetic parameters and see what happens in the morphology. And one of the things that this quickly revealed is that a continuous change in the underlying molecular parameters produces a discontinuous change if you look at it in morphospace. They called the morphological or developmental model a morphodynamic model because it has to do with the spatial interactions of the signaling molecules during development, and those interactions end up producing morphologies that are discontinuous. And so you see that over here at the right in the morphospace, areas of that morphospace where you just can't get to. So to summarize that, empty spaces, there's ordinary gaps. Everything's good. If we're doing morphometrics, if we're calculating a mean on those moles, I have no hesitation about that. If we did an ancestor reconstruction in that space, I think it's well sampled enough with real organisms and there aren't huge gaps in it. I'm pretty comfortable as a scientist with saying, yes, the ancestral morphology probably lies in one of those empty gaps. With Kendall curvature, that's a problem that we basically know how to correct in the morphometric literature. There are ways of doing the projection into morphospace. We can test it, and there's papers showing how to test whether curvature is an issue for your analysis. It's usually negligible for biological data sets, and that's because developments in genetics and etc. mean that biological shapes are actually pretty similar to one another, as we would see in the ordinary gap example. But if you do work on other kinds of objects like stone tools, I've done work with archaeologists, or simulated shapes, those shapes can actually be radically different in terms of Kendall space, and you would want to do a correction. And again, you can test that by comparing the distance between the objects in the full Kendall space versus one projected into a Euclidean space based on Procrustes distance. And if those are very strongly correlated or identical, then you don't have a problem with Kendall curvature, but otherwise you might need to take some precautions about doing certain sort of statistical operations if you're not taking that curvature into place. The Bookstein bend example is probably also pretty common. I showed you some examples, but any kind of phylogenetic structuring is at some level producing this kind of pattern because it's introducing some covariances and a gradation of similarities into it. If you are just looking at low principal components, some of those empty spaces, it's easy to overinterpret. You think, oh, there's a gap in the space. What does that mean biologically? And in fact, it may not mean anything biological. It may be an artifact of just the low-dimensional representation of a high-dimensional pattern. And statistically, even though we see that pattern in the low principal components, if you're doing statistics and model fitting on the entire morphospace, all the variables are all the piece principal components, in most cases you will alleviate those statistical issues. Although remember that high-dimensional spaces may introduce their own statistical problems with effectively over-parameterization. And if you were doing an ancestor reconstruction just based on one or two or three principal components, you may be reconstructing an ancestor in the bend rather than in the actual range of variation. But again, a high-dimensional version of that should avoid that problem. The constraint chasms, that's more of a real biological issue. That is really a question about whether the mathematical space and the biological space map onto one another. So a biological transition that's the easiest one or the shortest distance in terms of biological processes may not be the shortest distance in mathematical space. And all of our geometric morphometric spaces are mathematical. And it's essentially caused by a nonlinear relationship between the biological path and the mathematical path. And that may very well thwart ancestor reconstruction or model fitting, maybe or maybe not statistics. And this, I think, is a very interesting one. I don't have any answers to give, but I think it is a very germane topic for research. When do biological transitions not map onto the shortest mathematical transition is a question that we can ask. And a work that I have found really fascinating in this context is the work of Sean Rice, which is encapsulated in several journal articles, but also in his book called Evolutionary Theory, where he was really looking at the roles of development and nonlinear relationships between, say, genetic parameters and phenotypic parameters. And one can conceive from that work ways of thinking about genotype-phenotype map that combine with morphometrics could be used to model biologically realistic trajectories if we know something about the underlying development. And the paper I mentioned before that I wrote at Benedict Hallgrimsson's invitation was exploring that as an idea. And then finally, one thing that I'd like to plug here is a paper by Philippe Mitterrocker and Simon Hüttiger that's just simply about concepts of morphospaces. And this is a really nice paper for anybody who is working with different types of variables. Geometric morphometrics is purposefully designed to have the properties that I just described. But when you start looking at other kinds of variables, they may have, the morphospaces constructed on them may have wildly different properties, and therefore one would want to think about statistical models in a different way if one was using different kinds of morphospaces. It is not a one statistic fits all spaces kind of situation, and this paper explains why. Thinking forward, these are questions I've posed and given absolutely no answers to because I don't know the answers. But thinking about future research and method development, are there better ways for evaluating empty spaces, routinely testing for artifactual patterns like the Bookstein bends, and are they Bookstein bins, or are they constraint chiasms? Is this a biological empty space in morphospace, or is it an artifactual one? Are there better ways for managing high dimensionality? And this is a topic that many people, including many of the predecessors in the Rolf Medal, are working on. Trade-offs between an analysis in full versus reduced dimensionality. We often like to work with the reduced dimensionality because it reduces the number of patterns and we can see the patterns. But on the other hand, full dimensionality is capturing some of the, particularly avoiding artifacts like the Bookstein bins. Are there easy to use tools that can be developed that would test for these effects in this pattern on evolutionary models that could be employed by people who either don't want to or really don't have the expertise to really explore these problems in morphospace. And then finally, I'd like to say that most of the work I do, many people these days use R for various reasons, somewhat historical and somewhat preference. I use Mathematica. And if any of you are interested in using Mathematica, I have a number of packages, and many of the graphics that I've had in my talk today are produced in Mathematica using these packages, which you can get on GitHub. And then thank you to all the people who have worked with me, been my mentors in various ways, helped me learn geometric morphometrics. The ones in the corner have been particularly influential on me. Miriam Zeldich, Phil Gingrich when I was a postdoc at Michigan. Phil first got me from being a discrete character phylogeneticist to looking at continuous traits by giving me so many fossils that you actually had to measure them and look at variation, which I had never done before. Miriam Zeldich introduced me to a lot of geometric morphometric concepts. Norrin MacLeod, who was in London at the time that I moved to London, now with Nanjing University. I learned a lot about morphometrics from weekly conversations with Norm. And Jason Head, long-term collaborator, who in many ways has challenged my morphometric expertise by saying, could we do X? And I said, well, I don't think anyone's done X. And then Jason and I would brainstorm and find a way of doing X with morphometrics. And then others too. Amelia Martins, my former colleague at Indiana University on phylogenetic comparative methods, Andrea Cardini, who I've collaborated with a lot, Joe Felsenstein, who did a lot of the groundwork in phylogenetic comparative methods, but also has answered a lot of my questions when I've had them. Other collaborators, Katrina Jones, Ida Gomez, Steve Lacomber, my former PhD student Michelle Lawing, and also my former postdoc Anjali Goswami, and undergraduate Radha Komal, with whom I wrote a paper that until very recently was my highest cited paper, was her undergraduate thesis. So thank you all very much. I entertain questions in Stony Brook. I can't entertain them now, but you could always write to me. Thanks a lot. Bye-bye.